Okay, so everyone, thanks for your attendance. This is a truly international audience. We have visitors from Australia. I'm in France. I'm sure we have a contingent from Colombia and elsewhere in the region of the Americas. And of course, Joselin's at KAUST, eagerly awaiting to defend the outcomes of her research. So I'm not going to give a huge introduction other than to say that Joselin's been a key member of our team for the last 4 or 5 years. Joselin arrived in, I think it was late 2016, maybe late 2016. Gosh, that's gone quickly. To really fill a gap in our expertise in hyperspectral sensing. This was something that we wanted to move into. We hadn't done a great deal before. We had some nice instrumentation that hadn't been used and was sitting there idle. And Joselin came in and really kickstarted and grew that research in a way that I think few other people would have been able to do. And I think, as you'll see from the results of her research, she has really reached beyond the initial dossier of that research project to encompass a whole range of projects and explorations as they became available. Joselin's had a interesting background. She comes from a geodesy background in Colombia and has had a range of industry and government experience before that or prior to that, and is on the path now to a very exciting next step of the journey. She's just recently accepted a postdoc at NASA Goddard to work on a new mission there, which is great. It's going to expand upon the work that she's been doing here, really builds on the work that she's been doing here, I think, but open up to a range of very exciting possibilities and other research avenues at NASA. So hopefully before that, she'll be able to take a break, because I know the last several months and more have been extremely busy for her and a hectic time for everyone. So with that brief introduction out of the way, I'd just like to provide some broad ground rules for the defence here. The idea, for those who haven't attended it before, is that Joselin has an opportunity to explain her research in 45 minutes or less, thereabouts. How you encompass that from 5 years of research into 45 minutes is always a challenge, but that's the one she has ahead of her. The audience will then have an opportunity to ask any questions that they may wish on the various aspects of that research or other tangential topics. Joselin's done far more in this research than she's going to be able to present. So if there's questions that are somewhat related, pose them to her. The committee, who I'm very happy to introduce, you'll see on the display here, Professor Pei Hong from the Water Desalination and Reuse Centre. Professor Mark Tester from the Centre for Desert Agriculture, and our very distinguished external reviewer, Professor Pablo Zarco Tejada from the University of Melbourne. Pablo is a world-leading expert in hyperspectral sensing and UAV applications of those technologies, in particular for precision agriculture. So in terms of an external review, we really could not have found a more accomplished and high quality scientist to review this thesis. So Joselin, with that in mind, the floor is yours. I'll ask everyone to mute their microphones. The proceedings are being recorded, so keep that in mind with your behavior. So yes, again, if we could mute the microphones, everyone, that would be great, myself included. Joselin, over to you.
Okay. Thank you so much, Matt, for this introduction, and hi, everyone. I'm really excited to have this opportunity to share with you the results of my research in monitoring crop development and health using unmanned aerial vehicles, or UAVs, hyperspectral images, and machine learning. So let's start with a quick review on agriculture, one of the most disruptive activities on this planet that has been changing our relationship with nature. Since more than 10,000 years ago, when we were just hunters and gatherers, until today, when we are producers and consumers. We have domesticated different types of crops, expanding our croplands to occupy almost 13% of the global land area, advancing our agriculture together with our technology across our history. But recent studies are warning about the insufficient productivity and the unsustainability of these croplands, especially in light of some of the coming challenges like the projected population of 10 billion people by 2050, pandemics, economic recession, climate change, or the biodiversity collapse. So the question is, how in this digital age, when we have access to drones, images, sensors, machine learning, robotics, GPS, how using this technology we can bring real value to farmers and plant scientists to enhance our crops, to make them more resilient to different environmental conditions, and also under limited resources. Today we are going to cover 3 different cutting-edge technologies: hyperspectral, drones, and machine learning, and how to use them and integrate them into the phenotyping and precision agriculture pipelines. Let's start with a description of hyperspectral by comparing sound and light. Both are waves, which mean they have frequencies at which they oscillate. So the frequencies of sound determine the pitch that we can hear, and the frequencies of light determine the colors that we can see. So if we take a picture with a conventional camera like those that we have on our phones, an RGB camera, we will be able to capture 3 different frequencies: blue, short frequencies; green, medium frequencies; and large frequencies with color red. This is nice, but it will be quite limited if we want to get more information about the objects that we are sensing. So in this case, perhaps we should use a hyperspectral camera that helps us to collect hundreds of frequencies simultaneously, covering the visible and the infrared wavelengths. A hyperspectral image is a data cube of information that can be represented as an array of pixels with hundreds of layers, each of these layers comprising the information of a specific wavelength. If we fly a drone with a hyperspectral camera across the different phenological stages of a cropland, what we are going to get is a multi-temporal collection of hyperspectral images that contain the information of changing colors across the different phenological stages of the plants. So the incident radiation that reaches the leaves can be either absorbed, transmitted, or reflected. And the amount of energy that is reflected across the different wavelengths is what we call reflectance, and is what the hyperspectral cameras are able to collect. If we take out one of the pixels out of these hyperspectral data cubes and we plot the different reflectance across the wavelengths, we are going to get a spectral profile per pixel. And these spectral profiles provide information about different biophysical and biochemical traits, like leaf pigments in the visible range, cell structure in the near infrared, or about water content in the shortwave infrared. So now that we can see the applicability of hyperspectral in monitoring crops and plants, we can move to the main objective of this project, which was translating time series of ultra-high spatial resolution hyperspectral images into physiological indicator maps of crop development dynamics like chlorophyll maps or senescence maps by using machine learning techniques, considering the big data that we are collecting and also assessing the results. Well, to reach this goal, I follow a 4-step roadmap, starting with the data collection. That was performed in an experimental field from the King Abdulaziz University in the valley of Hada al-Sham in Saudi Arabia. As part of a field phenotyping experiment that we ran in collaboration with our colleagues from the Center of Desert Agriculture of KAUST. A total of 1,500 plants, 200 plants, were from a wild species of Solanum pimpinellifolium tomatoes, which is a donor candidate with beneficiary traits for agriculture, especially salt tolerance, were transplanted in this field of 80 square meters, and we monitored this crop across 5 different stages: establishment, development, flowering, fruiting, and pre-harvest. So 36 of the plants were randomly collected or selected to sense 3 of the top leaflets using a field spectrometer to collect the in situ reflectance of these leaflets, together with the relative amount of chlorophyll using a spot chlorophyll meter. Simultaneously to the in situ observations, we collect with a hyperspectral camera the 5 different hyperspectral data sets that we need for the retrieval. The information that collects this camera comprises the information from 400 nanometers to 1000 nanometers in the near infrared into 272 spectral bands. We fly a hexacopter 15 meters above the ground level to reach a 7 millimeters pixel size. Now that we collect the information or the data, we have to translate these images into, or process these images by calibrating the radiometric characteristics and the geometric characteristics of this image. To start with, I'm going to go through the geometric calibration. So the sensor that we use is a push room sensor that scans line by line the surface, or organizing the lines into single swaths, as you can see here in this figure. And to cover the full area, we have to collect several swaths to stitch them together to create a mosaic and to locate this mosaic in the right geographic position. To do this, we develop and implement a workflow that is based on computer vision techniques, starting with translating the raw images into luminance images, then applying different filters, horizontal and vertical filters, that help us to identify automatically key points at different scales, like corners, edges, or other details, specific details that we can use to stitch the images. And also, we have to describe the neighborhood of each of the key points by using different filters as well that help us to translate the color gradation around each of the key points into orientations. And this is what we can use as description to make these points unique. To georectify the hyperspectral images, we use an RGB-based mosaic, georectified mosaic, that we collect with a conventional camera that we fly simultaneously with the hyperspectral camera. So the key points are identified in both datasets, in the hyperspectral and the RGB base. And using the descriptors, we look for matches, but some of the matches are not good, so those are outliers. So we have to clean these outliers, and we follow an approach that is called maximum likelihood sample consensus, which is a routine that starts with randomly sampling the matching points to fit and evaluate a geographic transformation, then estimating the distance error that we reach with this transformation, and if the distance error is below a threshold of pixels that we define, in this case was 2 pixels, then our matching points are going to be classified or labeled as in layers. And we repeat these several times until we reach a good number of in layers. And these in layers is what we use to stitch the images together to create the mosaic and to georectify this mosaic. The full process is automated. We run this process in a parallelized computational approach in our supercomputers here in KAUST, and this saves us a lot of time that otherwise it would have taken us a lot of time if we tried to do this manually. So here we can see a comparison between the before and after the georectification, and I want to drive your attention especially to this irrigation pipeline. You can see that these mismatches between the swaths, and after the georectification you see the continuity of the pipeline. We assess these results by collecting some ground control points with GPS in the field, and we estimate the mean absolute error and the root mean square error metrics, reaching errors between 4 and 6 centimeters. And this is great because this is a fully automated method. Here you can see some of the results. And let's focus on this corner, the top corner. You can see some different illumination conditions between the quarters of the field. And this happens because to cover the field, we have to fly at different times, and sometimes the atmospheric conditions change. And this is one of the consequences of this. We have different colors, let's say, or different illumination conditions in the mosaic. To correct this, together with translating the original numbers or the original information that the hyperspectral camera collects in radiance units into reflectance, we have to perform a radiometric calibration of the images. We follow an empirical line approach that starts with placing on the ground a set of wooden panels with different colors, 4 of them with different degree of colors, and 1 of them fully white and the other one fully black. Then, using a field spectrometer, we collect the reflectance spectra for each of these panels, and we use this as a reference. Simultaneously, we sample the pixels in the hyperspectral images that correspond to these panels to extract a collection of radiance spectral profiles. They're going to be used together with the reflectance reference to establish or to fit different regression models that we can apply to the hyperspectral images to translate the units between radiance to reflectance. So here we can see some of the results, but you can see that the results are quite spiky. The spectral profiles of the hyperspectral reflectance images are quite noisy, so we have to polish these results to avoid any variations in further steps. So to do this, I apply a smoothing filter and also a denoising approach that is based on principal components that separates the noise from the signal, keeping the signal in the first components. To evaluate this polishing, then we sample some of the pixels that correspond to plants. And here we can see the spectral profile of one of these is in red color. And we evaluate these against some of the field spectra profiles that we collect in situ. And we can see that they align quite well. So this is, now that we know that our images are fully calibrated, we can use them to translate them or to estimate out of them biophysical or biochemical traits. In this case, our target was chlorophyll levels that we measure using the SPAT, so it's in SPAT units, by using machine learning techniques. So we use the in situ data to train the models. This in situ data, the target to retrieve it is the SPAT value, and the predictors that we use from this in situ data comes from the spectra, and each of the spectral bands is considered as a predictor. But we want to also analyze or train the learning of these models with some other type of predictors. So using the hyperspectral or the spectral profiles, we estimate vegetation indices to evaluate them as predictors as well. Vegetation indices are parameterized formulations that inform on a specific plant traits. One of the most common ones is the normalized difference vegetation index that is a direct indicator of greenness. It's also known as NDVI and uses the information of the near infrared and the red to estimate the level of greenness. Some other characteristics from the spectral profiles that we can analyze are the slopes. So one of the most common ones is the slope between the red and the near infrared. And this slope is well known as the red edge position because the maximum slope, the position of the maximum slope, is an indicator of senescence. And to retrieve this, we can estimate the first derivative of this profile, and the maximum point is going to give us the position. Some other characteristics or some other information that we can explode from the spectral profiles are the absorptions. The absorptions are a good indicative of the chemical components like chlorophyll in this absorption, or also water content in this other absorption. We are going to focus only in chlorophyll content here. And we apply a technique that is called continuum removal, which helps us to estimate the area of the absorption and also the depth of the absorption. Using this type of vegetation indices, we estimate 60 of them. Some of them inform on greenness, some others are about light use efficiency, some others are related with senescence, stress, water content, and of course, leaf chlorophyll. In this figure, you can see different rows, and each of the rows is representing one of the each vegetation index. The gray colors are indicating the spectral bands that we have to use to estimate each of these vegetation indices. And you can see that overall, we have to use the information between the 500 nanometers to the 800 nanometers that overlaps the maximum absorptions of the chlorophyll A and chlorophyll B. Using 145 bands in total, we estimate the 60 vegetation indices from both the in situ data and the hyperspectral images. And here you can see an example of the vegetation indices in a map form. Well, something important from the hyperspectral data is that this data is redundant, so is multicollinear. So this could be a problem if we want to perform retrievals, especially using regression models, because some models are not able to deal with this multicollinearity in the data. And this can be visible if we estimate a coefficient correlation like the Pearson's between different bands. So you can see especially high correlation between continuous bands because they are informing on similar frequencies or wavelengths. One way that we wanted to try in this experiment was to reduce this redundancy by translating the spectral bands into vegetation indices, and also in order to add interpretability to the model, since the vegetation indices are direct indicators of specific vegetation traits. Well, the machine learning modeling workflow that we propose and follow has 5 different steps, starting with defining the target to retrieve the spot or chlorophyll levels, and also evaluating 3 different sets of predictors. From one side, we try first with using all the hyperspectral spectral bands. Second, we try with the 60 vegetation indices. And then we try with the 145 bands that we use to retrieve the vegetation indices in order to compare if actually we are going to get similar results or better results with the vegetation indices than using the hyperspectral bands. In the second stage, we continue with the learning of different models. And for learning these models, we use the in situ data, splitting this data into 80% for testing and training, and the other 20 remaining for validation. So we train different regression models under a cross-validation technique with 5 folds that basically splits the training and test data into 5 different folds, using 4 of these sets to train and the remaining one for validation or for testing. And after running this routine, we estimate the goodness of fit of these models with the R-square metric. In order to select the most or the best performing methods, we run validation, a second validation of the models using the remaining 20% of the observation data, estimating the accuracy using the root mean square error and the mean absolute error. And then based on these metrics, the best performing methods are used to perform the predictions, feeding the models with the hyperspectral data cubes, and plotting or retrieving the spot or chlorophyll maps, and also retrieving the most important predictors. Well, considering that this data that we were using is multitemporal, and that we want to capture, model, and retrieve the dynamics of chlorophyll, we try with two different training strategies. From one side, the sequential, which trains all the models stage by stage using the in situ data stage by stage, and the retraining strategy that uses the data from the preliminary or the previous stage together with the current stage that we are training. In this way, we train 17 different models from different approaches, supervised approaches that are widely known and used in the machine learning literature. And the reason of using all these models is because we want to evaluate which of them are able to deal with this multicollinearity. So I'm not going into details how each one of them works. At the end, if you have some questions, I can answer. So one of them was the multivariate lineal. The other one was the partial least square, which is widely used in hyperspectral studies. The other one was decision trees, growing different volumes of leaves, ensembles of trees using two different techniques and growing different numbers of trees. And the difference between the random forest and the boost approach is the way the forests make the final consensus to retrieve the predictions. Support vector regression, which fits different polynomial functions in the form of kernels to perform the predictions, and the Gaussian process, which is a Bayesian-based approach that uses a prior of multivariate Gaussian distributions that we feed by using the observations, and at the end what we get is a mean distribution that can be used to perform the retrievals and also to retrieve the uncertainty of the retrievals. This is based on covariance functions, and these covariance functions measure the similarity between points to perform the estimations, and also in the form of kernels. Well, after we train all these 17 models, we perform a comparative assessment of the different metrics that we estimate, the goodness of fit and the two metrics for accounting for accuracy. So here in this figure, we can see a summary of the results. And overall, we have three conclusions. From one side, most of the models, when we're trained using the vegetation indices as predictors, reach similar goodness of fit and accuracies than when they were trained using either all the spectral bands or the 145 bands. And this is great because it means that we can actually use vegetation indices to reduce the dimensionality of the data without losing information and reaching similar accuracies. The second conclusion is that under the sequential training strategy, most of the models were able to reach a good fit or goodness of fit, but than in the retraining case. But under the retraining case, we reach higher accuracies than under the sequential strategy. Based on these metrics, we select some of the best performing models to analyze how these models if these models were able or not to capture and model and retrieve the chlorophyll dynamics. So one easy way to visualize this was by estimating the average of the predictions at a plant level, and here representing each of the averages per these pixels in this box. So each of the boxes represent a specific time, and we compare different models under different training strategies and also using the different predictors. We include in the comparison the linear model just to showcase that the linear is not a model that is able to deal with the multicollinearity of the data, and this is visible because the retrievals are very poor. Whereas the other type of other models like the random forest or Gaussian process or PLSR perform very well and are able to capture the chlorophyll dynamics, especially under the retraining strategy. You can see how during the first stage, the models are retrieving low chlorophyll levels. Then in the second stage, the chlorophyll level increases, reaching a peak in the third stage. And then because of the aging process, the chlorophyll level decreases again. So overall, what we can get from here is the first PLSR, the one that is widely used in hyperspectral, has a good performance only under the sequential training and using all the spectral bands. Whereas the random forest and the Gaussian process were able to perform and retrieve very good retrievals under most of the conditions that we evaluate. For the Gaussian process, we were able also to retrieve the uncertainties, and this is great because we can have an assessment or an additional metric of assessment of the retrievals. A third criteria to analyze the results was also comparing the validation, the distribution of the validation data that is represented in this figure with the gray delineated boxes against the distribution of the retrievals. And what we can see overall is that the PLSR distribution under the sequential training strategy using all the bands together with random forest under the retraining strategy and the Gaussian process under the retraining using vegetation indices are the most similar distributions. Based on all these assessment criteria and metrics that we evaluate, we choose as the best performing models the PLSR, random forest, and the Gaussian process. And we fit these models with the hyperspectral images to plot or to retrieve the spot maps or the chlorophyll maps. And here we can see an example of some plants across time and the comparison of the results between the different models. And also for the case of the Gaussian process, the retrieval of the uncertainties. In addition, we were able with these three models to retrieve the most important predictors, either if spectral bands or vegetation indices. Here I'm showcasing only the random forest case with green colors. You can see the most important spectral bands. And this is great because we can trace back which spectral ranges are playing a relevant role in the retrieval of these chlorophyll dynamics, and also which vegetation traits represented in vegetation indices are playing an important role in chlorophyll, the retrieval of chlorophyll. A summary of the most important vegetation indices and spectral bands identified either by 1, 2, or the 3 models is shown in this figure. Well, now with these results, we can use these results directly to make decisions in precision agriculture activities. But if we want to use these results for phenotyping purposes, we have to dig a little bit more into these results and produce more insightful metrics out of this. So we have to perform some analytics with these retrievals. This is the fourth and last stage. We showcase these analyzing the plant senescence and salt stress dynamics. Something that I didn't mention before is that 2 of the quarters of the field were irrigated with salt water. We applied 5 different salt treatments with different salt concentrations in the field. And also the other 2 plots in the field were irrigated with fresh water for control. Using the Gaussian process, we retrieve the chlorophyll levels of the total population of the plants, and in this figure you can see somehow to get an insight about the distribution, multitemporal distribution of the retrievals. However, it's not that visible in using the chlorophyll content as a trait. It's not that visible the salt stress effect in the plants. So we want to evaluate as a second trait the growth. And to estimate the growth, what we did was to, in an automated way, delineate the plants and measure the area, the projected shoot area per plant. And here in this multitemporal distribution comparison between plots, you can see actually the salt stress effect in the S1 and S2 plots. We can also retrieve, or we also retrieve the uncertainties of the predictions. We reach similar uncertainties across time, except for the second stage. These uncertainties were between 5 and 10 SPAT units, and for the second stage, below 15 SPAT units. Well, since we want to analyze senescence, especially the salt effect in the plants that were irrigated with salt water, we have to keep in mind that the senescent process in these plants has two components. From one side, the natural aging process of the plants, but from the other side, the salt-induced senescence playing a role in this process. And we want to account only for the salt-induced senescence. And to do this, we can use this index, the salt-induced senescence index, that is reported as well in the phenotyping literature. And we can do it per accession. Well, something that I didn't mention before is that we use 200 different accessions. One of them comes from a commercial species, the Heinz. This is the species that is used to make ketchup. And the rest of the species, the rest of the accessions come from the Solanum pimpinellifolium. From a preliminary study that performed our colleagues, the plant scientists in the salt lab, different levels for 6 of the accessions, different levels of salt tolerance were established. High tolerance, medium tolerance, and low tolerance for these 6 different accessions. And for the commercial one, of course, then the tolerance is null, and this is quite evident here. The plants didn't survive until the end of the experiment. And we use these accessions as reference. Well, to analyze the senescence dynamics, we translate or we reclassify the chlorophyll levels into 3 different classes: young, senescent, and healthy. And here we can see a zoom in of some of the plants and how the distribution of young, senescent, and healthy pixels can happen within the plant. To estimate the salt-induced senescent index, we have to measure the amount or the area covered by the senescent pixels per plant per accession at each of the stages under salt and under control conditions, together with the projected shoot area per plant per accession at each stage under salt and control. And once we measure this, we can establish the salt-induced senescent index per accession per stage. One way to visualize this and to analyze this index that we found it was the best was by performing some data visualization techniques using a scatter plot with a marginalized histogram that showed the relationship and the distribution of the salt-induced senescent index across accessions between salt treatments at 3 different stages. So each of the plots is divided into 4 quarters. The first quarter contains or comprises the accessions that receive a high impact from the 2 salt treatments that we are evaluating in this stage. In the second quarter, we have all the accessions that receive a higher impact from the second salt treatment. In the third quarter, we comprise all the accessions that receive either a lower impact from the first stage, or from the second stage, or that recovery from the first salt treatment. And in the fourth quarter, we have the accessions that receive lower impact from both salt treatments. We evaluate these across 3 different stages, and what we can conclude here is that in the last stage, in the 4th quarter of the last stage, we could have some candidates with high or with good qualities for salt tolerance. Another pattern that we analyze using the accessions that I showed before that we use as reference is that the closer these accessions are to the origin of the plot, the higher the tolerance level is. And we analyzed this across the time, and the pattern was stable. So we can use the radial distance as a secondary indicator, or as a secondary index to indicate somehow the tolerance, the salt tolerance in these accessions, together with the quarters. To evaluate these results, we analyze the different traits, the different indicators like growth, senescence, against economically primary traits like biomass and yield, reaching high and significant correlations between them, especially during the last 2 stages, pre-harvest, and under control and salt conditions. And in addition, the last analysis that we ran was using 2 different indicators reported as well in the phenotyping literature that account for salt tolerance and stress tolerance using biomass and yield. And we wanted to evaluate if there is some correlation between these 2 indicators and the salt-induced senescence index. What we found is that actually no, there's no significant correlation between them. But and perhaps we shouldn't use this salt-induced senescence index as a direct indicator of salt tolerance and stress tolerance. But if we dig a little bit more and we analyze the salt-induced senescence in a different way, in a multitemporal way, and stage by stage, and also comparing the different salt treatments, the performance of the accessions during the different salt treatments, what we get is that actually the quadrant and the radial distance indicators are highly positive and negative correlated and significantly correlated with salt tolerance and stress tolerance. Well, and this is the end of the road. I just want to share with you some other publications that I did during my PhD. In the corner here, you can scan this code if you want to access to the list of the publications. But here I'm just going to show 3 of them. One of them was using a time series of hyperspectral satellite images from a commercial farm here in Saudi Arabia. So what we did was to use a spatiotemporal kriging model to fill the gaps that we can have in these images, like cloudy areas. This was a collaboration with NASA. And we used this time series of hyperspectral images to retrieve leaf area and also chlorophyll content running a cubist regression, which is a regression model similar to random forest, evaluating as well vegetation indices. And this project was led by one of our former researchers, Dr. Rasmus Hoberg. And also, I have a collaboration with one of our former students to evaluate a random forest using vegetation indices and hyperspectral bands in a species of with Halim, our former student. Well, let's move to the conclusions and directions for further research. About the data collection, what I can say is that using ultra high resolution, like millimetric or centimetric resolution, we can explode the weeding plan information. But this is going to increase exponentially the computational effort and the storage to perform the different process that we have to go through. But at the same time, a benefit out of it is that we can use the purity of the pixels, the pure spectral signatures that we can get from each of the pixels, because we are reducing adjacent effects. As a direct, we can use them directly to retrieve leaf level vegetation indices. One suggestion that I will propose for the research would be like performing sensitivity studies to evaluate and determine the best pixel size accordingly with the crop, or accordingly with the structural characteristics of the plants that we are going to target. For the second stage, for the processing, of course, it depends on the sensors that we are using, but in general, I suggest the use of or the integration of inertial and navigation sensors like RTK sensors in the payload that will help us to collect in real time accurate coordinates, geographic coordinates that we can stamp on the hyperspectral images. And also, it will be helpful to use solar irradiance sensors to measure the upward and downward radiation, either installing the sensor in the payload or in situ, to collect these during the scanning, and in this way enhancing the radiometric calibration. For the modeling step, the approach that we propose here, the framework that we propose here, can be used with neural networks, with convolutional neural networks, with deep learning or reinforcement approaches. It's important to look for dimensionality reduction when we use hyperspectral data, not only for reducing the computational effort, but also in order to identify the prediction, the importance of the predictors, and also to add interpretability in this way to the model. And together with this, looking for models or for approaches that help us to measure the uncertainty of the estimates. These two things will help us to open the black box that sometimes machine learning is. I would encourage the use of vegetation indices predictors, because with these we can explore the reflectance features like absorptions, maximum and minimum points, inflection points, the slopes, that are sometimes are direct indicators of a specific trait in the vegetation. The use of alternative strategies for modeling time series of data. It's important to capture, model, and retrieve the natural dynamics of the traits that we are targeting. And I will suggest, in the case of Gaussian process, the use of spatiotemporal kernels or spatiotemporal covariance models that account for the spatiotemporal autocorrelation and stationarity in the data. And finally, it's important to run comparative evaluations between different models, because at the end, machine learning is a data-driven approach, so it will depend as well somehow in the data that we are collecting. So we have to try with different models and see which one of them are able to deal with the data that we collect. For the last stage, we see how it's important to explode the retrievals if we want to integrate this information and insightful indicators into the crop modeling, phenotyping, and genotyping pipelines, not only with the retrievals or the vegetation indices, but exploring them, exploding the spatio-temporal dimensionality, the within plan variation. Is important to explore it to retrieve secondary metrics like or health indicators like the salt-induced senescence index. Two general conclusions that could be sometimes, well, could be sound obvious, but it's something like sometimes we want to capture a lot of information or a lot of data in the field without having or without being clear about which specific traits we are targeting. And this could, if we know this from the very beginning, we can save a lot of time at the end an effort in the field, and also we can get a better idea about the retrieval strategy that we should use. We can use either machine learning, vegetation indices, or radiative transfer models, or combine them to perform our the estimations. And finally, establishing transferable modelings or modeling frameworks is very important. Here I show a model or a machine learning framework that can be used under different data sets. So we just need to change the data set, feed this framework with the new data set, and perform the retrieval. So it's important to establish more than fitting different models. The importance is relies on establishing these frameworks. Well, finally, of course, all this effort couldn't be possible with the help and collaboration of a lot of people. So I want to acknowledge our team, as led by Professor Matthew McCabe, my advisor. Also, Dr. Rasmus Howard, who was at the beginning of my PhD mentoring. Samir, for all your effort in the field and the logistics. Stephen, Joanne, thank you so much for your collaboration. Casper, Oliver, the students that were working with us in the field as well, collecting this data. Alejandra, Halim, Bruno, Mateo, Christian, Omar, Ting, and Dan, thank you so much. And of course, my special thanks go to the Salt Lab, led by Professor Mark Tester. This was an amazing collaboration, an amazing experiment. Thank you to my dear colleague, Mitch Morton, Dr. Sonia, Gabi, thank you so much as well for your effort in the field and in ARA. Professor Magdi Musa and his staff in the King Abdulaziz farm. That was an amazing work in the field as well. And finally, last but not least, Professor Arco Lucier and Dr. Darren Turner from the University of Tasmania, who helped us and collaborate with us in the calibration of the hyperspectral data. Well, thank you all for being here with me, for attending this presentation. To my family and my friends that are in Colombia in this moment, it's 3 a.m. So, muchas gracias por estar aquí atendiendo esta presentación. I will be happy to answer and go through your questions if you have some.
Fantastic, Jocelyn. I think we could all take a moment to virtually clap or physically clap if you've got your video there.
Nice.
Wonderful presentation. I think from that overview, you can see that Jocelyn's really mastered not just the remote sensing, but the spatial statistics, programming, big data analytics, and the one that probably I find most important, the graphic design. I love the presentation and the graphics that you've done over the years. It's a really nice way of communicating these results. Okay, so the next and most important part of this oral defense is now the questioning of Jocelyn. The way that we do that, as I mentioned before, I give the audience an opportunity to ask Jocelyn some questions, and then the audience will depart the room, and the committee will then have their turn to focus some inquiries to Jocelyn. So how we might do that, given that it's a virtual forum, is that we will I'll look at the participant list, and if you can raise your hand, I'll unmute you and you can ask your question directly, or you could type your question directly into the chat, and I'll do my best to transcribe that to Jocelyn. So you Shan, I know you have a question there already. Excellent. So how about you unmute and fire away.
Hi, thanks Jocelyn for your great talk. I have some question for your radiometric calibration. It seems that you calculate the images directly from radiance to reflectance. So do you use a model to fit the reflectance radiant to reflectance without the simultaneously irradiance measurement? Because I know that when you collect the images from the UAV, that you have you need to collect all the data during a period of time. So how does the radiance variation contribute to noises to your data?
Okay, yes. Well, thank you for your question, Yushan. This is precisely one of the things that I try to skip to don't go into the details here, but there is something that I didn't mention from this calibration, and is that actually the hyperspectral camera what captures is digital or it stores in the hyperspectral images is digital numbers. And there is a set of calibrated calibration that we perform in the lab that we can run after collecting the data to transform the digital numbers into radiance units. So this is first. So then the radiance units are translated into reflectance. But to start with, from digital numbers to radiance, we use this set of parameters that we establish in the calibration in the lab. And second, of course, somehow the spiky results that you can see here are affected, of course, by the field by the different irradiation conditions that we have in both data sets, because although it was kind of simultaneously the collection of both the reflectance spectral profile and the hyperspectral images, it was not exactly the same. So this would introduce somehow this effect as well. And also because we are performing regressions, so it's part of regression model to have some uncertainty. So I don't know if with this I answered your question or.
Yes, I think that's probably it. And also, I'm interested about you say that you use the principal components to separate your signal and the noises. So basically, that you say the first component represent most of the signals. So just wondering that how much variance the first component represent to all of your data.
Yes, I'm going to just to go to the slide. So here in this is what you are talking about, yeah, about the minimum noise fraction. So this method, as I said, is based on principal components. So the first components, the first 6 components, this is so a pattern that I found when I was calibrating all the images. The first 6 components were those that keep the most information or contains the most information. And after this, the first these 6 components, the 7 and so on, these components have a lot of noise, and you can see these. We can evaluate these only visually, but also seeing the high variability of the histograms or the digital values of the pixels. You will see these if you just evaluate quickly, you will see this. So of course it depends on the data that you are using, but at least in these images that I use, we are keeping the most information in the first 6 components.
Okay, thank you very much.
Okay, I know we're going to have some questions from the Halo group for sure. Casper, you'll have a question, I see.
Hey, yes, first of all, thanks for a great presentation, Jocelyn. That's really truly impressive work you've done. So very, very, very impressive. So in your kind of framework, you have obviously tested quite a large number of machine learning approaches. So I was wondering if you were to kind of repeat the study or do a similar plant experiment or even mapping a different crop, would you recommend to generally just stick with those machine learning approaches that you recommended in this study, or would you recommend to try them all out to see which one works the best?
Yeah, that's a great question. So I would recommend just using those that we know that these are not based on non-linear regression, because actually we see that it's very obvious from the multivariate linear that we are getting very poor results. So we can skip this one, no? The use of non-linear models, we can skip them. Even under support vector regression, we shouldn't evaluate linear kernels, or the Gaussian process has also this option of evaluate linear kernels. So I would suggest just skip this. Perhaps only using ensembles of trees instead of decision trees, single decision trees, it could be a best practice as well. Because with ensembles of trees, we have a consensus and a better distribution of, a better evaluation of the different predictors between these different trees that we are growing in the forest. So I would suggest go for non-linear. In terms of the decision trees, go for ensembles of trees, either random or boost. And for support vector regression and Gaussian, try with different kernels, yes, but non-linear kernels.
Okay, thanks very much.
Okay.
Okay, I have a question from Rahul, just a quick one while the other members of the HALO group compile their questions. What was the signal to noise ratio of the sensor?
Okay, good question. Well, it is not stable. Let's say it is not. We don't have a single value for this, like an average or a mean value for this. But what we did is in the calibration, as part of the calibration that we run in the laboratory, what we found, you can go for publication that we have with one of the master students, and Casper, was that we evaluate the signal to noise ratio across time and measuring several times in situ and also in the lab to see if it was stable. So somehow it's stable, yes, but it will depend on different. I mean, it will depend on the way you are calibrating the sensor as well. So I don't have a number. I can't say it is 1 or 0 or 100. I don't have a number of these. But the recommendation is this: performing preliminary calibration of the sensor to evaluate this and to see if the sensor is performing well and you are getting good signal out of the sensor before going to the field to run a campaign.
Okay, Yosin, it seems that you've dumbfounded the audience. They've answered all of their questions, one or the other. I'm going to go for the latter. With that in mind, I'd take this final opportunity to thank Yosin, for the audience to give their appreciation, and then I would also ask them to quickly vacate the room so that we can continue on with the next phase. So Yosin, thanks for the great presentation.